stuff I have, Okay. So one of the challenges of getting into uh, a new area is that you might be thinking, okay, this is great. I can kind of, I can see myself sending samples. I can see myself getting data back. I can see my results. And then you're like, oh, then I would have to write a paper or communicate to somebody what that this whole experiment is about. And that can be a little daunting. But so that's kind of the purpose of these types of workshops. And we also we're talking about we talked about collaboration and letters of support. We also provide methods so that you can be able to have those methods that you can put in a publication, so that when some reviewer asks you something specific, and I get these emails sometimes where people are like, can you help me answer this reviewer's question, because this person's like really geeking out about the mass spectrometry and I don't know. It's always reviewer two, isn't it? So we're going to spend some time talking about just the kind of like looking at them, what the difference is between DDA, or data dependent acquisition, versus DIA, versus TMT. And yeah, she's getting that there. And so just at one level, I would say DDA has historically been better for high sample numbers compared to a TMT experiment, because TMT stands for Tandem Mass Tag, and that means you have an isotopically an isobaric label, a label that has been designed, engineered to have only specific carbon and nitrogen isotopes in there, and then you actually label each sample. And so that number of tags limits your experiment. And so DDA, if you had, 30 samples or more, then you would go with DDA. Well, now DIA is kind of, in the last 5 years or so, is kind of become very relevant as far as quantitative proteomics. And it also is good for higher sample numbers. But it's the same thing. You're going to reduce an alkylate. We haven't talked about reducing and alkylating, but that's one of the standard things we do that Karen's going to talk about here in a minute. Adding trypsin, getting your peptides, and then getting MS1 and MS2 information. The only difference in the TMT experiment is that the only difference, there's several differences, but we're going to make a bunch of tags that we're going to label covalently bond onto every single peptide in a certain sample, a tag that we can then knock off later that will tell us the identity of which sample that came from. And then your eyes are not deceiving you. We're going to take all of those samples after they've been labeled at the peptide level. Some of those are controls, some of those are treatments, and we're going to combine them all together. Whoa! So now we get this multiplexing sort of advantage in that now that one peptide that was in one sample, that same peptide that's in the other samples, they're all going to come out at the same time. And so you're going to get a higher response for each peptide. And you're only measuring it one time. Instead of measuring that peptide in sample 1, and an hour later, you're measuring that same peptide in sample 2, and an hour later, you're measuring that sample peptide 3. And then, you go on, you have this instrument maybe changing as time goes on. In TMT experiments, you're measuring that peptide one time for all your samples. And so there's a lot of stuff. It's a much more difficult experiment to do because you have to deal with these isobaric labels. It requires offline fractionation. So we have to actually use an offline fractionator to take that one sample and split it up into multiple samples that we then combine again. And then we use that tribrid mass spectrometer that we just showed you the guts for, and we get an MS1 signal, an MS2 signal, but then we take some of those fragments, collect them, hit it with higher energy, knock off those tags, and we measure the MS3 signal. Whoa. And the height there gives you the quantitation. we actually just measure the difference in heights here and know that the 126 tag went to sample 1 and the 127 tag went to sample 2, et cetera, et cetera. So then DIA comes along and it says, wait a minute, this whole thing here is all built upon looking at the highest, most abundant precursor, and then the next one, and the next one, and the next one. Let's just do it differently. Instead of that, let's take all of the sample and let's have a quadrupole that will divide up the sample into windows. So let's say 12 Thomson windows. So all of the peptides that are between 400 to 412 M over Z will get isolated by the quadrupole, and those guys will all get fragmented at exactly the same time. So now it's like if I took a piece of glass here and every one of us put our thumbprint in the same place, right? And then I took a computer and each one of us put our individual thumbprints into there. And that computer had to go back and look at that mass of thumbprints and try to pull out layer by layer whose thumbprint was in there and who wasn't. Okay, it's kind of like that, except that the retention time is going to, for each fragment, is going to follow a different pattern. So if you look at it the same way you look at DDA data, it looks like a mess because you have a bunch of fragments from a bunch of different peptides all colluting at the same time. But they're not, because what happens is that if you put time in there, all of the fragments that are with, say, the blue peptide, represented by the blue peptide, are going to actually have a different envelope. They're going to elute they're going to colute together. And then you're going to, for the purple and the red, you're going to have the same thing. And so being able to put time back in there, whereas an MS2 up here in the DDA is a very static snapshot where we say, well, that's from one ion, and we can figure out what the spectra is, the fingerprint for that peptide. Here, we would say, well, this is one fragment, that's one fragment, that's one fragment, all of the same peptide. And so once I know the M over Z of those fragments, I can figure out what my peptide identity is. Okay, so just to go into a little bit more detail and make it a little bigger so that you can see it here. This is just the same slides, just bigger here. The dirty word here for DDA is stochastic. The stochastic sampling means there's some randomness to the DDA. You might have, in some of your samples, your low-level guy, you might see it in some samples, but in other samples, it just didn't catch it because there's some other things that distracted it, okay? And this thing tends to favor the higher, most abundant peptides. And again, going from biological information to digital information. For the TMT experiment, it's still a DDA experiment. We just label your samples on the front end with tags, and TMT is a thermo product. There are other, was it iTrack or whatever? There are other people that do labeling, but Thermo's pretty much taken over the labeling experiment. And they have a 10-plex, they'll have a 16-plex, they even have a 32-plex now, which is really double 16 sort of. But the problem with these plexes is that the molecule that you're adding onto your peptide, once you get from 10, when you go to 16, the molecule is bigger, which now it becomes greasier. It's more organic in nature, and it will stick on the column longer. And we don't like the TMT 16-plex as much as we do the 10-plex. And there's some other downsides to that. But essentially, you can take these plexes and you can put them through your offline peptide fractionator, run them on your Orbitrap clips, and then the database search that we'd use is an open source program called MaxQuant that you could download on your computer today, and you could do that, and it will search. And the great thing about our TMT is that we can do proteomics with our TMT. So we would, and Rick's going to spend more time talking about that. Phospho for DIA has come along, and we are about to the point of being able to do those projects as well. But from that data set, out of max quant, we can do normalization and differential analysis like we can for all of our things. So again,
short question.
Yes, sir.
So on a 10-plex, does it mean like 5 replicates of a control and 5 replicates of a treatment? That would be 10-plex?
That would be it. That's right.
Thank you.
So you got the MS1 signal, the MS2, and again, to get from MS1 to MS2, you add energy in there. That's that collision energy to break apart one of these guys into, you isolate it, and then you hit it with energy, and you break it apart into its fragments, and then you hit it with more energy to actually get the quantitation here, which makes you might think then, well, how do you do the quantitation for DDA? And that's a really great question. Without TMT, do you quantitate on the MS1 level? Do you quantitate at the MS2 level? Do you count the number of times that you see that spectra? What do you do? And that we could have a whole weekend on that as well. Just to talk about fractionation just for a second, just so you understand what I'm talking about, I'm talking about using another LC system that's not hooked up to a mass spectrometer, that has a UV vis detector. And we put in the sample, and we would fractionate it, and we collect the sample back. So we have to use a non-destructive detector to be able to do that, because a mass spectrometer, once you put your sample in, it's gone. And so, and then for DIA, we used to say this. We used to say that DIA was really good for serum and plasma, and for IPs, bio-IDs, and things like that, because they weren't as complex as, say, like a cell lysate. And the great thing about DIA is it's equally sensitive to low abundant peptides. But we now say that DIA is really good for everything, because the instrumentation is faster, that we can do all of the, and the software that is searching, the database software searching it is better. So let me just show you what DIA fragmentation looks like. So, and I should have rewrote this, but this is for this peptide A, G, F, whatever. And then this is what the signal looks like. And it up here, it's telling you, I rewrote that at least, that you've got your Y6 ion and your Y7 ion, and then these are some of these other ions here, or like there's a Y4 down here, and they're all co-eluting at the same time. And so we're using this information, which ions are co-eluting here, that match up to the fragmentation pattern for that particular peptide. So that's identification, but then we're also looking at the quantifying by drawing a sort of a Gaussian curve across there. So then the question in DIA has to come with, well, how many points across the peak do you have? And this is where we get all side-eye at Thermo, because when they go to sell an instrument, it's not the engineers that do this, it's the salespeople. They'll do the settings, so it's really great of identification. So they get 12,000 proteins in every run, but they only have 1 point across the peak. How are you going to quantify with 1 point across the peak? You don't. So you got to count. Now I'm literally saying you got to count 1, 2, 3, 4, 5, 6, 7, 8. So we have about 7 or 8 points across the peak. And so there's this envelope here where we're going to do our quantitation. And I borrowed this from Brian Searle. I was up at US HUPO a couple of months ago and sitting in one of his classes, and he had this great little chart here. So here's what happens if we oversample. If we take too many samples, then we're basically using instrument time that we don't, we're wasting instrument time. If we undersample, we get a misshapen peak here that doesn't really accurately represent the quantitation that we want. So we want to find that Goldilocks best fit right in the middle there. And they think it's somewhere here in the 7 to 8 range. The guy that we worked with for years here from OMRF, Mike Kinter, he is a targeting guy. He was very much more comfortable with the 15 to 20 points across the peak. So we always had this running joke, because when Rick comes in here in this afternoon, he's going to say 5 is fine. So anyway, that kind of tells you, but this is the guy that helps us with the DIA. So Rick will talk more about the Astral mass spectrometer, but it has such a high sampling rate that it's able to go, instead of 12 Dalton windows, we can do 3 Dalton windows or 4 Dalton windows or sometimes 2 Dalton windows, and still with the right chromatography from we can still see that. And so this has been just a couple years old, and it's really helped us out a bunch in that. Okay, last thing before I finish, which I know I'm way late, but Karen doesn't mind, is that DIA analysis right now for us, for 40 samples, we're sitting at about $68 per sample. So for 40 samples, it's $2,720. That's our price. And so you could carve that up. That could be 8 groups of 5, 5 groups of 8, or 4 groups of 10, or however you like. TMT, and that includes everything. That's doing the digest and running the samples. Some people want to save a little bit. I think you save $23 per sample if you do your own digest.
And if there's tissue, like if it's a tissue, you're
Thank
gonna
you.
have an extra added.
That's right. If you're sending us hunks of tissue and we have to do the tissue lysis, then it's going to be another $23 per sample. Thank you for pointing that out. TMT analysis for 10 samples, because the cost of those kits, it's about $2,000 there for 10 samples, for 10 labels. And then when we do our phospho TMT analysis, we actually, you get two. You get a regular total peptide experiment, what we call the lysate experiment, and then we do an enrichment, and then we also do the experiment for the phosphopeptides. And so this experiment's a little bit unique because it'll have, basically you get one set of data that's at the protein level, and then you get one set of data that's at the peptide level, the phosphopeptide level. But that's helpful because you want to be able to know, is the reason why my phosphorylation is going up, is it because my protein expression went up? That would, or not. And so you get that in there. And then we're going to talk about the output that you're going to get later, but these are all things that you're going to get. Oh, we do. Yes. An online question.
You can go ahead next. Unmute.
Did they raise their hand? Oh, cool.
You don't have it set where they put.
Oh, hi. I have a small question. So could you go back to the price part? So for the DIA service, it will, if we digest the samples by ourselves, it will be $27. And if you do the whole survey, the full service, it will be 68 samples, $68 for one sample.
You mean if you do the digestion or you do the lysis? Which are you talking about?
The DIA part. So if we do the digestion, it will be 27, or is for the TMT. I get confused here.
Oh, yeah. So the run is $45 per run to do the data collection.
Okay, so it's not including the sample preparation, everything. It's just the run.
Yeah, 45. Yeah, so you're going to pay $45 per run for data collection, and then we charge, in this slide here, showing you that the triptych digest is another $23. So if you were to do your own digestion, then you would just pay $45 per sample.
Oh, so if you do all the service, it will be $68.
If we do the triptych digest and do the data collection, it's $68 per sample. But what Karen was saying is that if you send us tissue samples, and we have to do a lysis on those tissue samples, since that requires more, then that's another, you say 27? $23 per sample on top of this.
Oh, okay, $23 plus 68. Okay, got that. Thank you so much.
You're
Sorry
welcome.
to interrupt you.
No, it's fine. It's great. That's a great Sam question too. Sam's great at all that. So we've covered a lot of stuff here. There's a lot of things in this map here, and I loved, you're like, what's going on in your mind? You have like a question like that, or anything else here. And I can't reiterate this enough, that where do I start? You always start with a conversation with this man right here. He is very responsive by email, and he has been doing this for, I don't know. He was the core before there was a core. He was the first guy when we had one instrument, and he did everything. And now he specializes in being our front door, and he does a lot of conversations with people. Answering these questions, addressing, figuring out what your needs are, and everything. So, and we're going to try to get him up here for lunch or something. He's down there working, collecting samples, doing stuff. But that's where you start, is with an initial consult, and those are all free. And one time he let us know how many conversations that he's having with people. And he put that up on the board. And I mean, he's having 40 different conversations going on all the time, emails coming back and forth. And so, and that's, he's got email chains and all of that. So I just want, it's not just like, oh, all I need is an address and a price, and I'm just ready to send samples and be done. We have people that come into proteomics that way. That's a little bit of a painful way, unless you have learned some other way. The real way is to learn by having a conversation first and gaining from that. Any questions before I hand this over? Sample prep. Yes, sir. Ryan, and then we'll head over there.
Can you guys do other digests other than tryptic digests, like cytosine and stuff like that?
Yes. So Sam would talk with you about that, but we've done like ASP-N, and there's been a couple that we've done. Sometimes we might suggest you could do the digest on your end and then just send us and tell us. And then when we do the data, when we do the search, we can just tell the data searching software that we have a different thing. But sometimes we'll buy that and do the digest here.
And we can also go ahead. And we can also send you protocols, like the protocol that we would use to do the protein purification and things like that before you do the digest.
Is the selection between DIA or TMT be dependent on the research question, or we can start either way?
It does. So I think what Rick would say if he were here is he'd say, if you're just starting off and you have this paper and you say, hey, I want to do TMT, we would talk you out of it. Unless there was a specific reason. Really the only customers that we have that still do TMT is because they've done TMT for a while and they don't want to switch. But yeah, I mean, that's a great question to have with them right now. If you're going to go phospho, though, that has been the reason why we still have the TMT.
Good. I have two more questions. With regards the sample, I'm working with whole blood plasma samples, and we have a variety of good plasma for start off, from really expensive tubes until, the regular EDTA tubes that we can use. What would be your recommendation? Should we start comparing those samples between the same participant, for example, and then choose our best option for analysis, or should we use regular plasma samples, for example?
So you're asking about, you've got these different collection tubes and you want to be able to use all of them, but so I think you would have to do some sort of small scale comparison first. Yeah.
Yeah. Okay. And my last question.
Promise?
Not for the day. If I am interested in a particular set of, for example, proteins related to lipids, can I see that without doing like a lipidomic profile?
Are they proteins that do they have lipids on them? Or are they just proteins that like to hang out with lipids and do lipidy-like things? Mm-hmm. Then I would say we don't need to do lipidomics to do that.
Good.
My understanding is lipidomics would be looking at proteins. When I hear that word, I would think that you're looking at proteins that specifically have lipids hanging off of these proteins, and you're wanting to know that information.
Yeah. Thank you.
Uh-huh. Yes, ma'am.
So you have this new shiny piece of equipment, right? So I presume that you still have the older ones. So the pricing that you're showing, is this only for Astral? Are you still, no?
No,
So this is for,
the Astral is a different price. So that one,
okay.
so if we go back here, this price says for the
Oh, Orbit, oh, sorry, I
Explorer's
missed the,
class.
yeah,
Yeah.
I thought that you, okay. So Astral will be more
But-
expensive.
You can do the 20-minute run on the Astral, which is just the shorter run.
Okay,
For the same price.
okay.
Yeah, so did you guys hear that? So this is, it doesn't say here, but this is an hour's worth of Explorer's time. Costs the same amount as 20 minutes on the Astral. It's that good.
So are you gonna give us like, some sort of a list of all the instruments that you have and, different options that we have so we know what is the best for?
To our webpage, down at the bottom, there's a price list.
I see.
But yeah, we can talk.
It
Okay.
kind of gives you,
Yeah.
there's the Astral and the Eclipse, we don't have anymore. I need to remove that off.
Okay,
the-
okay. Thank you.
but We'll get to that. One.
Okay, well, ladies first, and then we'll head back there. Okay, Divya.
So because the tryptic digestion breaks the lysine residue, when we are looking at ubiquitination, the ubiquitin chains are connected at the lysine residue. So will that affect it?
It does. It does. The trypsin doesn't like to cut where there's been ubiquitination.
Yeah.
So we have what's called our miss cleavage rate should be about 10% or so. It goes up to 50% when we do ubiquitin samples, which when we're like, okay, so that means there's been a lot of ubiquitination. That's great. We're seeing a lot of that.
So is there an alternative digestion technique?
Well, I don't think that the ubiquitination occurs at the arginines. So you still have those. And as long, so if you're not seeing what you could go to a different thing. I think the other thing we do is to really make sure that the trypsin works well is that we include Ly-C. Have you ever heard of trypsin Ly-C? So it's like trypsin, and then it's like really, really good trypsin because it will like, I think its claim is that if the lysine is near a proline, then it doesn't want to cleave, and the Ly-C will cleave there as well. So I think that's what we would suggest is doing that. But again, these are all great SAM questions when you have your consult with them. But yeah, Alexia.
Two short questions. So some proteins undergo proteolytic digestion naturally or during sample prep, and that may not be on a lysine residue. Is the database takes it into account, or those peptides will be lost?
Takes into account
the cleavage that is not on a lysine. Arginine.
Yeah, so that's where you get murky, because if you have non-tryptic cleavage, splicing or cutting there, we need to tell the database searching software to look for those things, which essentially takes and makes it from an easier task to a very complicated task, because now it's like, oh, well, then all this noise here could be a cleavage point here, a cleavage point there, a cleavage point there. So we tend to say, we can though come back and do what we call, I think there's a couple of different versions of it, but it's like a semi-triptych where it'll say, okay, we're going to look for some of that.
So it's feasible, but you need supercomputer for that. Right?
Well, it's just, you're going to end up getting a lot of information that you're going to have to sort through.
And the second question, for the quantitative proteomics, spectral counting, is it something that to use instead of TMT?
So spectral counting would be, if we just did a DDA experiment, the question is, and we haven't gotten into this, is how do you quantify that peak? Do you quantify it at an MS1 or an MS2 level, and how do you do that? And so what people did back in the day is they would just say, well, I'm just going to count how many times I actually see this particular M over Z, And then that's going to be my counting measurement. The problem with that is that's assuming the chromatography is the same, right? Because as the peaks widen because of bad chromatography, then you're going to see it more. And so I would say spectral counting is okay, but it's not it's really just for DDA. We'd rather have a way of quantifying that's more robust, like intensity at the MS2 level. Not to say people still do spectral counting, and there's a way to actually normalize those, and that's fine. It's just that's a legacy way of doing it. Can I put that in a nice way?
You're saying it's an old technology? Yeah. Okay. Mm-hmm. So TMT is the new one?
So TMT, basically by having those isobaric tags there, that is a much better way to quantitate at the DDA for a DDA experiment. And then DIA can actually quantitate at the MS2 level, and that's a really good way. Yeah.
Thank you.
Mm-hmm. All right. Are we good? Are we ready to get Karen up here? Because you know what she's going to talk about? Sample prep and what you should be asking at the consultation and all of that.
Yeah, so actually Dennis already went through a lot of